This is episode 21 of the Immunology Podcast, Trained Immunity with Dr. Mihai Netea. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast. We have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. Today we have Dr. Mihai Netea from Radboud University Nijmegen on the podcast to talk about his research on the memory traits of innate immunity. We've also got our usual roundup of recent highlights in immunology news coming up, but first,
STEMCELL Technologies would like to introduce you to human immunology news. It covers everything from immunotherapy, autoimmunity, and adaptive and innate immunity. Human immunology news keeps readers current with the latest news, research, policy events, and jobs relevant to the immunology community. Subscribe for free at www.humanimmunologynews.com.
all right, Brenda, so you're not calling us from the Netherlands. You're out partying somewhere. Where are you at?
Yeah, I'm way down south in my hometown in Argentina. So that's also maybe the connection is not great. I don't have all my gear.
You sound a little bit robotic today, so we'll apologize to the audience. But okay, Brenda Robot is fine. It can be Brenda 2.0. So are you still on holiday then? Does this mean you have like an extended, super awesome holiday?
Well, I just started. This is Wednesday. So I've been having 3 days of holiday so far. So
Okay.
it's nice.
Did you work during Christmas?
Up to Christmas, I did. Then I took a week off and I went to Italy. Okay. Yes. And sometimes I take holidays. I'm sorry about that.
So you had a holiday in Italy and then a holiday in Argentina, but you were trying to say your only holiday was in Argentina because Italy is not far enough away from you for it to count as holiday.
Yeah, that's a European way of life. You take holidays and enjoy yourself.
I got back from Hawaii recently, so I did get to do that.
It was pretty nice. How was it over there?
It was good. It was quite the flight. It took 21 hours. Layovers, 2 kids. Thank God for tablets.
But I'm glad you got to enjoy some sun. I think we should move on to the reason we're talking, which is exciting science of 2022.
So Omicron.
I don't want to talk about Omicron. Oh my gosh. It's everywhere. I just cannot take this anymore.
I've described it as the Oprah Winfrey of viruses, where you get Omicron and you get Omicron and you get Omicron and you get Omicron. Everyone's getting Omicron.
It's wildfire. It's everywhere.
I'm still waiting. I'm sure it'll happen.
I got a couple of close calls. I was very concerned. I was not going to be able to get into a plane because of my failed PCR test or something.
Quarantine somewhere. Fun time.
So exhausting. I do not have an Omicron. I already exhausted my Omicrons on the last episode. So this time I have something else. But you do have something Omicron related, don't you?
Oh, I have no. Well, so, this is a paper that's out. We'll just jump right in. So this paper's out, which means it's about it doesn't even have the Delta variant in it because it takes forever to publish, folks. So it was received on April 21st, 2021, revised on April 16th, 2021, and accepted on the 4th of January, 2022.
That's insane.
Yes. So the paper is mRNA 1273, aka Moderna, vaccine-induced antibodies maintain FC effector functions across SARS-CoV-2 variants of concern, which are no longer the variants of concern. First author is Paulina Kaplonek. Last author is Galit Alter. It is an immunity. This is still a preprint. It's not even technical. It's printed. It's not a preprint.
But what's the variant of concern back then?
Alpha. But Omicron shares some of the same mutations as alpha in the spike protein, folks. Has the same mutation that caused the S gene drop on some of the testing that we do. So it's relevant because it's not like these mutations are all de novo in every variant. All the variants are compilations of different mutations that make it a distinct enough entity that it keeps propagating, and we see some of the same mutations pop up over and over again. So this looks at spike protein mutations. Long and short, what they go into, and I'm going to keep this quick, is that if you look at antibodies that are generated by vaccines or infection, they have several functions. One's neutralizing, right? It binds to the spike protein in such a way as that it prevents the virus from binding to a cell and entering. That's neutralizing antibodies. That's neutralizing immunity. That's what a lot of people measure. But they said, wait, antibodies do a lot of other stuff. They have a lot of FC component, right? Functions, complement mediated, phagocytosis, targeting cells for death in other ways. There's a lot of other functions that they do, right? And so this paper compares natural gained immunity versus vaccination, looking at isotype and subclass binding in terms of neutralizing antibodies, in terms of FC receptor binding, the ability of the FC receptor to bind it, right? So low affinity IGFC receptors binding, the ability of receptor binding domain. So that's the part that binds into the cell, right? So that's really neutralizing antibody, but in kind of like a fragmented form. And then FC of function, FC effector functions. So let's see here. Let me get the whole list because they did a whole bunch of things. Complement deposition being the main one that they looked at, and antibody-dependent phagocytosis. Yes, so both of them. So they look at all this and basically find that, yes, Moderna's vaccine is compromised in neutralizing antibodies with some of the variants, although not nearly as much as natural immunity. So natural immunity is the worst. But the ability of all these other antibody functions to work is largely preserved after Moderna vaccination, whereas natural immunity, not so much. And how do they do this? They take plasma from people like 10 weeks after their vaccination dose. So pretty, not that soon, but in their peak. So pretty good, strong antibodies, and then compare it to people post-infection. So, and then they compare and then do all these studies. It's a really short paper and it gets to the point. And there we go. So the vaccines have other antibody functions that work just fine.
Okay, I guess that's a relief. That's a good thing.
Everyone's measuring neutralizing antibodies, but forget antibodies have other functions.
Yeah, that's actually, it's always difficult, right? To measure all the layers of function of the immune system with something that's simple, replicable and applicable at a large scale. So it's nice to see that they're looking at different functionalities of antibodies.
So good paper, short and to the point. I'm surprised it took so long to come out.
Yeah, I wonder why. What kind of obscure issue happened there? But I guess that I can continue with my first paper of the day. I don't have a COVID paper, but I do have a vaccination-related paper. And I want to start with that one because it is in some way a follow-up from one of the papers I discussed last time, or about the idea of aerosol vaccination through the airway. And I was very excited about this paper because it comes from the same group from McMaster University in Canada that is developing a nasal vaccine for COVID, which has recently started a phase 1 trial in December. And they actually published already the results from a phase 1b trial with a vaccine that is against tuberculosis. And I thought that was very interesting because maybe us living in developed countries may forget that tuberculosis is still quite a widely spread disease. Pulmonary tuberculosis in adults can be a huge burden in certain parts of the world. And I didn't know this, but according to the WHO, there's about 1.4 million deaths a year that are related to pulmonary tuberculosis. And I thought I was not aware that the numbers were still so high. And also, so we know that we get vaccinated when we're very young with BCG, and that protects us against this childhood tuberculosis. You will think, but does not really protect us from a pulmonary tuberculosis in adults. And so there's a huge interest in trying to develop a vaccine that can give us long-lasting protection against this part as well. And I guess as the same with COVID. It makes sense to make a vaccine that is inhaled. And so these people have this paper was published in JCI. The first author is Mangala Kumari Jayanathan from the labs of Zhao Jing and Fiona Smile from McMaster University, which is in Ontario. And it's titled Aerosol Delivery, but not intramuscular injection of adenovirus vector tuberculosis vaccine induces respiratory mucosal immunity in humans. So I think I'm already giving up a lot away from from the content of this paper. They have this vaccine that has a antigen 85A within a serotype 5 adenoviral vector, and they test humans a regimen in which they compare either what they consider a high or a low dose, which is actually quite low when you compare with what they give in the intramuscular injection, which they also compare in this study. And they look specifically into mucosal cells in the lungs. So they do lavages and they obtain bulk from these patients, and that's how they really look into T cells that are in the mucosa of these patients. And they have in total 31 participants, so it's not a huge study, but it is supposed to be a phase 1. They show that they can successfully deliver a sufficient dose of this vaccine, that the patients can really tolerate it very well. And when they look they look a little bit closer into the cells in the airways and also the cells in the blood of these patients. And they find that after the vaccination, for patients that had aerosol vaccination, they see an increase in immune cells in the airways, which is not the case for intramuscular vaccination. They see that a really robust increase in CD4 T cells are specific against this 85-8 antigen, and that many of them are polyfunctional. They express interferon gamma, TNF alpha, IL-2, and they're really the dominant T cell that gets elicited in these patients. They also see a smaller but consistent increase in CD8 T cells, which is interesting because they also see a small increase with intramuscular vaccination of CD8 T cells, but not for CD4s. And then they show that these cells have the markers of tissue resident memory cells expressing CD69, CD103, and that the vaccination is very specific against the antigen that they are targeting, because although these patients these patients have a pre-existing immunity against because of BCG vaccination in their childhood. So these patients can respond to a cocktail of mycobacterial antigens even prior to vaccination, and all the other responses to other mycobacterial antigens stay the same, and only against these 85A antigen is that they see increased responses. And so I thought it was really interesting because they show that there's a very specific increase in the mucosal response, and that and they also see an increase in T cells in the blood, so it can also use vaccination. And they always see that the dominant T cell that gets activated are CD4 T cells that are dominating the immune response. What else they show is that although all of the patients had pre-existing antibodies against the vector, the human adenoviral 5, they actually don't have such high. They measure IgG titers against this virus in both the circulation and the BALF. And what is interesting is that they see they have indeed neutralizing antibodies in the blood, but in the BALF titers are much lower. And I think that's really good, because I think this type of vaccination needs to be the future for diseases that are acquired through the airways. And I'm really looking forward to the results from their COVID vaccine study that started in December, and so I'm really looking forward to see what the future brings.
All right, we're gonna go on the next one. This one's called Behavioral Immune Landscapes of Inflammation by first author is Georgina Hranehus, and last author is Andreas Hidalgo. It's in Nature. It came out the 5th of January. It is an interesting paper that in some ways is really what you would assume and is obvious, but no one's ever done it before, which is why it's cool. So you have different immune cells you can sort by flow. You can then sort these cells into subphenotypes based on RNA-seq and single-cell RNA-seq data. And so you can try to figure out, well, this cells we expect to do this, and these cells we expect to do that. Well, what they did is they used intravital microscopy to create morphogenic patterns of cells. So instead of doing like a single-cell RNA-seq, they said these cells in these situations behave this way. They're this shape, this size, they move in this manner, they're this directional or non-directional, they're asymmetrical in this ways in their shape, they're blobby, they're gooey, they're, little podocytes. They use all the different ways that you can use mathematical framing to describe these cells and put it in n-dimensional space and then map out the differences between these cells. So, and then they used inflamed skin and trachea as other mechanisms as well through like an ischemia model to then look at pathologic perturbations. And then what they did is they did behavior screening in 24 different mouse mutants that were related to a specific pathway, which I'll get to in a second. So at a high level, they were trying to say, okay, let's pick one pathology. So pathologic neutrophil activation, over activation of neutrophils. So they looked at something downstream of a receptor called PSGL1. And so they said, okay, this is something we know. We looked at this one and noticed there were pattern changes. So then they scored for 24 different mutants that were downstream of this signaling pathway that weren't like a priori already known to work, right? They were trying to figure out if they knocked out one of 24 different mice. And they did the same experiment in 24 different mice, not knockdown, knockouts. Did they see a shift in the pathology in this morphogenic trait that said, oh, this is why this gene here is a precursor effect for what you would expect to have downstream? So they looked at intravascular neutrophils, intravital microscopy, and what they did is they looked for behavioral reprogramming. And so they found this gene FGR, which is a Src kinase not associated with altered inflammation generally. And its deficiency does not compromise neutrophil recruitment to sites of inflammation. But they did intravital microscopy and found big behavior shifts in one of the populations of cells that they defined as a bad actor. And then they looked at a therapeutic targeting of it by looking at neutrophils that cause early vascular activation and damage. So you use their models. And then did pre-inhibition of this pathway ahead of time prior to injury. And also did the knockout. And so what they're getting at here is if you did single cell RNA-seq, you wouldn't have necessarily, and you wouldn't, regular population wouldn't know. So you'd have to screen a whole bunch of mutants. But then if you would have to screen all these mutants, then do the final assay and say ischemia in the mice for all of them, right? And do all the time points and have a big enough group to see that it had an effect on the pathology. So think about it. They did 24 mutants, but they didn't do 24 mutants with 10 mice per group doing ischemia. They did 24 mutants and took a couple of mice each and did the intravital imaging to figure out which of these had something funky in its behavior of one of these patterns, because they identified different patterns saying, oh, this pathogenic pattern is no longer there. It's gone. It's shifted to a non-pathogenic pattern. And then they go, now we're going to do the 10 mouse study on that one. And then they found a target. So it's a really interesting proof of concept. I don't know if it's quite ready for like worldwide showtime because they had to take 24 mice and do intravital microscopy on it and do very complicated mathematical modeling to essentially isotype these different mice. It's a heat map with 73 morphokinetic parameters that are all defined, and then they put those into 3 behavioral patterns. So it's a little user hostile, but you could see that if they could automate this in a way or have predefined programming for this, you can make this a less specialized process, and it can be very useful because it's really, instead of going, oh, well, guess what? If, structure dictates function, or, genetic structure or knockout or whatever you're expressing dictates what cell you are. Duh. What if we look at the functional morphology of it, isotype those, then monkey with stuff and see what goes wrong at that level, and then go to the larger system model of pathology. So that was a really interesting approach. It was a ton of work to do this, like incomprehensible amount. And kudos to them for that. And it's a really unique setup. I mean, they advertise in the abstract more than 100,000 reconstruction of cell shapes and tracks over time were required for this data set.
No, I did have a sneak peek at the paper, and it's super cool. I also really like the differences between the traces and a bit of these 3 groups of neutrophils and how they find these behaviors. I thought it was really cool, but man, so much work.
Yeah, it's impressive. So I don't know if it's quite ready for prime time. But all right, Brenda, let's get on to your paper before we start having to use your dial-up modem.
Yeah, so my last paper also has to do with neutrophils a little bit. So, and platelets. So also reminds me of some of our guests talking about the importance of platelets and neutrophil behavior. So in this case, I think it was a very interesting paper published in Immunology from first author Valentina Poli from the lab of Ivan Zanoni at Harvard. And it's called Inhibition of Transcription Factor NFAT Activity in Activated Platelets Enhances Their Aggregation and Exacerbates Gram-Negative Bacterial Septicemia. And the first thing that came into my mind is, what's the point of a transcription factor in a cell that has no nucleus, like a platelet? So I was kind of intrigued at the beginning because it feels like an impossible thing. So I looked into this paper. I thought it was really cool. In the study, they look into the phenomenon of septicemia that it's induced by gram-negative bacteria, and basically some of the events that happen when you have bacterial in your bloodstream. It's the induction of nets of neutrophil extracellular traps for neutrophils together with activated platelets. You have also intravascular coagulation, you have inflammation. And so in this paper, they look into how NFAT expression in platelets affects this, the activation of neutrophils and coagulation and basically the septicemia response in mice. And also they have a little bit of human data. So basically, they look into NFAT. As I said, NFAT is a transcription factor, and it's a little bit counterintuitive to look at it in platelets. But NFAT members are expressed in platelets, 3 of the 4 NFAT members. And it's activated by downstream of phospholipase C and by influx of calcium. And it binds, or it interacts with calcineurin and gets dysphosphorylated, and that's how it becomes activated. And they see that indeed in the case of platelets, upon the activation of platelets, they see that there's a time-dependent phosphorylation of, in this case, they look into NFAT1, one of the 3 members of the NFAT family are expressed in platelets. So they see that NFAT does get activated in this type of cells. And they also show that in the case, if you inhibit NFAT activation, and they have a couple of ways of doing this. They either have NFAT knockout mice, and they do in vitro experiments. Or they also have what I think is very interesting is what they call a vivid permeable peptide, which is a peptide of with this sequence that can inhibit the interaction between calcineurin and NFAT. and that results in an accumulation of phosphorylated NFAT, so a non-activated NFAT. And they see that this deactivation, this inhibition of NFAT results in a hyperaggregation of activated platelets. And this is true both for mouse and human platelets. So there's something that NFAT activation is doing to the behavior of these platelets. And in the case of NFAT negative mice or NFAT knockout mice, they also see that their platelets aggregated more efficiently. And this is related to some integrins that are enhancing the spreading of this platelets and fibrogen, which is part of their aggregation mechanism. And they also look into NFAT inhibitors, which are used as immunosuppressants, for example, for transplants, and to which altered platelet function had been reported before. The examples are cyclosporin A or tacrolimus, and they also drive hyperaggregation of mouse platelets. But this only happens when the platelets become activated. And they also look into a couple of patients from some syndromes that are characterized by hyporesponsiveness of platelets, and they show that, and they looked at inhibition of NFAT activation with, for example, cyclosporin A, increase the aggregation reached by these platelets, and they didn't try it in vivo, but they suggest that this might improve slightly the function of the platelets in these patients. And what is interesting is that when they have mice that are treated with, in this case, tacrolimus, also an NFAT inhibitor, they see that this leads to increased aggregation of platelets, which reduces, for example, tail bleeding, which is a way of looking into coagulation in mice. But what is also interesting is that they have an exacerbated pulmonary thromboembolism. So this mice have a higher tendency to coagulation, which will be kind of the other side of the coin for this treatment. And they look a little bit into what is related. They try to understand how does NFAT inhibition affect the platelet function, but they don't completely get into the mechanism. But then they develop a mouse which they call infatuation mouse, which is a transgenic mouse that is expressing this vivid peptide I mentioned a little while ago, which prevents NFAT activation by preventing the binding to calcineurin, and they have it expressed under the Cre recombinase, and then they cross these mice with PF or Cre mice to express this peptide exclusively in platelets. Basically, they see that it's NFAT expression specifically in platelets, which have no nucleus. So it has to be something that's not related to transcriptional function of NFAT that is increasing the aggregation of these platelets, and is kind of influencing their function. And they also have a tendency to form more complexes with neutrophils and induce the production of nets by neutrophils. So I thought it was very interesting because they study kind of a non-canonical function of NFAT, which is not related to transcription. So it was a really nice paper, I thought.
Did they figure out what NFAT was doing that wasn't transcriptionally related?
No, they kind of have that as an open end. They do suggest that maybe it's the fact that dephosphorylated NFAT might be kind of capturing or turning off some regulators of platelet activation, or maybe releasing something that turns off the platelet response. So they do not know exactly how the mechanism works.
Sounds like that's their next grant.
Yeah, I do think so.
Well, we're going to be speaking to Dr. Mihai Netea at Radboud University Nijmegen in just a moment. But before we get to that, you do immunotherapy research? STEMCELL Technologies offers products and protocols for immunotherapy research, including T-cell isolation, activation, and expansion reagents. Use easy-step T-cell isolation kits to isolate highly purified T-cells in as little as 8 minutes. Follow up with immunocult reagents designed for human T-cell activation and expansion. Learn more about stem cells' optimized protocols and reagents for immunotherapy research at stemcell.com/T-cell-therapy.
Joining us today is Dr. Mihai Netea. He is professor of experimental medicine at the Radboud University in Nijmegen in the Netherlands, and also professor of immunometabolism at the University in Bonn in Germany. The Netea Lab has made contributions to our understanding of trained immunity, which are very valuable, and we're very much looking forward to talking about these today. Dr. Netea, thank you so much for joining us.
Thank you for the invitation. It's a pleasure to be with you today.
All right, so I'll dive right in. Reading your papers, it's not often that you get to read some scientist's work and then it becomes a coffee table conversation at dinner. It's not like I can go talk to my parents about a lot of things I read. But your stuff was very interesting at a high level, the fact that there is generational immunological memory transfer that's epigenetic. So could you dive in at a high level and just jump into what you found?
If I might just say, maybe Dr. Netea could start by talking what do I understand as trained immunity?
Absolutely. I'll try with that one first, and then we'll go to the intergenerational transmission of traits. Basically, what we mean by trained immunity is the capacity of innate immune cells to change, to adapt to a previous encounter. So for more than the last half a century, starting from the 50s and 60s, we started to divide the immune responses into two. What is happening in the first couple of hours and days of an infection? These are processes that are mediated by certain immune cells called myeloid cells and NK cells, which are generally believed to react the same way each time that they encounter a pathogen, a microorganism, and so on. And they are supposed not to have memory. They were always doing the same thing. However, when the infection is longer, cannot be eliminated by this first line of defense, then other smarter cells come into play, like lymphocytes, B and T cells, which can, first of all, induce a response in an antigen-specific way. So they respond only towards very specific microorganisms, but then also they can build immunological memory. And we always thought that this immunological memory can be built only by lymphocytes. But in the last couple of years, we discovered that actually also the myeloid cells, NK cells, so cells of the innate immune response, what is happening in the first hours after an infection, they can also build a certain type of memory, which is not that specific as that of lymphocytes, but still very relevant for the next time that they will encounter an infection, when they will respond better, basically. So that memory, this heterologous form of memory in the innate immune cells, we called it trained immunity. Now, regarding the other question is, well, is that relevant only during the lifetime of an individual, or can be also transmitted some form of it to next generations and so on? Well, in the last, let's say 1-2 decades, more and more data have become available that indeed transmission of immune traits can be done also across generations. I think that by far the most solid information on that comes from plants, for example. If a plant is infected with a pathogen, with a virus or a fungus, and it is not killed by that pathogen, it will become more resistant. But very interestingly, this resistance can be transmitted through seeds, through epigenetic processes for up to 4 or 5 generations. So in the plants, this process has been demonstrated for quite some time already, actually. Now, people have started to ask themselves whether that could happen also in animals. And again, in animals, and especially in invertebrates, such as insects, worms, and so on, there has been demonstrated very clearly in the last 2 decades that there are more than 15 groups of animals, if I'm not mistaken, where this happens. And in others, it has not been studied. So basically, for example, an insect which is infected by a virus can transmit this resistance to the progeny, so the progeny would also be more resistant to the virus. And lately also there are more and more studies showing that similar type of processes are probably taking place also in vertebrates. And this is, I think, something very important, because in the end, that demonstrates that transmission, intergenerational transmission of traits has an important role in physiology. So yeah, I think that is very important as well.
So to continue on that, I think if I remember right, in your work, you showed it transmitted to F2, but not past that. So, the parent with the insult passed on some level of protection, and even across organisms, the one generation, and then it persisted to a second, but then waned off. That to me sounds like there's evolutionary pressure to lose the memory because it expends energy. Have you guys looked into that? And then what makes this memory fade over time? Is it just energy expenditure? Why is it 2 generations, not permanent, or not 5, or if there's any difference in between species that gives insight into the persistence differences?
So as I mentioned, in plants, for example, it's up to 5 or 6 generations. So it's absolutely clear that there is a difference between species and different types of organisms. I do think that there is indeed a pressure in a way to be flexible, to be able to change. Because what it has been good today or tomorrow doesn't mean it will be good in 10 years or 100 years. And for example, trained immunity, the capacity of responding better to a reinfection or a restimulation, is lost not only across generation, but is also lost within an individual. So for example, if we induce trained immunity with a certain type of vaccines in a healthy individuals, that doesn't mean that healthy individuals will retain that increased responsiveness of the immune cells the rest of his life. Actually, we do see that after 2 or 3 years, that starts to fade, and it is lost. And it is lost because this type of memory is induced by different programs at epigenetic and transcriptional levels in the bone marrow progenitors of immune cells. And when different type of infections takes place or different type of exposures are encountered by an organism during his or her lifetime, that will change basically these programs in the bone marrow. So this is a very flexible, it's a very plastic process. Is not rigid in the way that, well, let's say that we were infected with microorganism X, and then we will be able to better respond to microorganism X the rest of the life. But that might be not so good when we will encounter organism Y or a different one. So I think we are meant to be flexible, and we are meant to be plastic in this, because on short term, it makes sense actually to have this increased responsiveness, to have a changed program, but on the long term, not necessarily. And there is also the question, if this is good against infections, why didn't evolution take care in the hundreds of millions of years since we evolved, that we are always at the very high level of immune response all the time? We will not have any infections, everything is fine. Well, there is a trade-off in this, and evolutionary speaking, there are 2 things that impact the success of a species. First of all, not to die before you reproduce. And the second one, the number of children you will have. And for example, in our case, as vertebrates and mammals, the reproduction of the success of the reproduction is crucial, because we have very small number of progeny, basically. And for example, a woman who has a baby, when she's pregnant, she needs actually to tolerate a transplant, basically, because the baby is half coming from the father. So it's basically a transplantate for 9 months. And because of that, the immune system has to be tolerant, has to be at a lower pitch, actually, than we would need, for example, when we are when we are healthy and we would fight an infection. So there is this trade-off, and it has been shown actually in earlier studies that organisms, not only humans, but in other mammals as well, that if you have a very strong immune response, you do have a higher chance of spontaneous abortions. So all the time there is a trade-off: very strong immune system against the infection, but at the same time not strong enough to allow the pregnancy to be successful. And during a pregnancy, the immune system of the woman is physiologically depressed a little bit. So, for example, that's why in the clinic we advise women not to eat blue cheese, for example, because that is full with fungi, which in a depressed immune response, a physiologically depressed immune response, can give much more problems than in other situations. So, yeah, that is why we need to be flexible, and we need to change to be able to change our immune response.
Going back a little bit into the features of any trained immunity, I am fascinated by the fact that there's certain, for example, BCG vaccination can offer protection against diseases that are not related to tuberculosis, and how that also starting from epidemiological data has also sparked an interest in understanding how just training the immune system in general can protect us against a wide range of diseases, and also including COVID-19. So I would like also to hear your thoughts on this part of trained immunity.
So yes, it has been shown for a number of vaccines, BCG being one of them. Bacille Calmette-Guerin, the vaccine against tuberculosis, has been shown to induce heterologous protection against mortality in young children. So the mortality in countries with high infectious pressure has been decreased by 50% in various studies by BCG vaccination, but it's not the only one. Other vaccines that can induce these type of effects are the measles-containing vaccines, such as MMR, but also oral polio vaccine has been shown, and also probably variola vaccine was doing the same in the years when it was given. So yes, that has been shown, and it has been shown to decrease the number of other types of infections. We have done also a number of studies on that and showing this effect. That being said, we have also to realize BCG by itself cannot be the golden bullet for everything. So, for example, we have tried to use it also for COVID-19, and there are a number of clinical trials ongoing. Some of them, a couple of them, are known now. Unfortunately, it seems that BCG does not protect against COVID itself. It does protect against other respiratory tract infections, but not against COVID-19. On the other hand, there is another study published recently as a preprint, and hopefully soon it will be published as a peer-reviewed publication, with a phase 3 randomized trial with measles-containing vaccines with the MMR, in which there was, if I remember correctly, approximately 40-45% protection with MMR. And there is another very well done, very large study done with a vaccine against shingles, Shingrix, for example, which reduced by 20 or 25% the hospitalizations with COVID-19, and that was in a very large population, in 400,000 people or something like that. So there are these non-specific effects that have been seen also with other types of vaccines in COVID-19. It seems not with BCG, but probably with measles and Shingrix, so the shingles vaccine, and probably there are clinical trials also with influenza vaccine. But what we believe it's important to learn from this pandemic, because fortunately now we have good vaccines. So for COVID-19, we won't need BCG or MMR because we have better vaccines. We have a number of vaccines which work perfectly fine. So we would not need this heterologous protection. But by showing that this is happening also in this pandemic, these positive effects, we demonstrate that such heterologous vaccines could be used against another type of new pathogen. So for example, if we would have a new pandemic in the future, and we will have a new pandemic in the future, we could immediately try to use and employ in the very first months into a pandemic to do a couple of clinical trials to assess which one of these vaccines would work against that new pathogen. So at the moment that we would have that information, we could use it as a bridge vaccination until the specific vaccines are developed. Because now we were lucky, actually, to be very honest, that within 1.5 years, we had several good vaccines against COVID-19. But there are sometimes pathogens for which is much more difficult to develop a vaccine. We can see what is happening, unfortunately, with HIV. Still, after 40 years or so, we still don't have a vaccine against HIV. And we might get less lucky next time. So using these approaches to have the possibility to create a bridge vaccination until we are able to develop a new vaccine could be very valuable, I think, in the future. And we need to use the next couple of years to develop this methodology also for the future pandemic.
So I wanted to dive into this with mechanism a little bit. And by the way, I believe there was a paper that actually showed that flu vaccination was affecting COVID or vice versa. I can't remember now. But
yeah, correct.
There was this cross-link. It was pretty recent. But to deep dive just a little bit, do you think this is because you're developing a level of immune tolerance so that your body doesn't freak out and have a cytokine storm? And so the more vaccines and exposure you have, kind of the better muscle the immune system is? Because we all think about, it needs to be trained against a pathogen, which is true. But half of all the pathologies, as a physician, that comes from these diseases or more is an overreaction of the immune system to the insult. It does a great job killing the pathogen. It also just takes you out too, because it hasn't kind of learned how to calm down a little bit. Do you think that's what's driving it? Or is there another, or is it really a lot of cross reactivity and stuff, or some of both?
Well, I think you actually explained the precise correct answer in your question, because there are precisely these two components of the protective effects. On the one hand, the cells are able to react better in terms of eliminating the organism when it is encountered, but at the same time, it's learning not to overreact with a systemic inflammatory response. So both these two components are crucial, and we see it, for example, with influenza, but also with BCG vaccination, that indeed both of them decrease the systemic inflammation after they are being given, while improving the responsiveness while we encounter a new pathogen. So these two components are both very useful and very important. This is how the immune system actually learns to respond properly to an infection, to be very effective in eliminating the infection, but also tolerizing the products of that infection and not overreact to induce collateral damage.
And also, diving deep into this topic, what do we know about the mechanisms by which you can generate this trained immunity? I know that you have some insights on how these vaccines can affect the metabolism of monocytes and train them. Maybe would you like to give a quick view of what do we understand at this level?
Yes, so there are several levels at which we can understand that. So at the molecular level in each cell, what is happening basically is an interaction between immune signaling, which is induced inside the cell upon recognition of the pathogen. So the pathogen is recognized through specific receptors that induces certain immunological signals in the cells. And those signals go at the level of the nucleus and at the level of metabolic changes in the cell. So it changes basically the metabolism of the cell. It's inducing certain pathways such as glycolysis, is changing other pathways such as Krebs cycle. And that results in a different mixture, in a different soup of metabolites in the cells. And those metabolites are at the same time those which influence thereafter epigenetic enzymes in the nucleus to change the chromatin architecture. And then there is this interaction between immune signals, changed metabolite concentrations in the cells, and modified epigenetic enzymes, which change the architecture of the chromatin, which makes it more appropriate for gene transcription to respond easier to stimuli when encounters infections and so on. So this is one level at the molecular level. At the same time, there is also the cellular level where this happens, and this can happen at cellular level at periphery, in the monocytes and macrophages in periphery. But very important also, this happens also at the level of the bone marrow progenitors of these cells. So we encounter these changes that are induced by the vaccines also at the level of the bone marrow, where the myeloid cell progenitors are changed by these signals. So they will produce thereafter daughter cells, which are better in eliminating pathogens.
So we're running out of time to deep dive science here. So I guess the question I have next to kind of go to is for the audience listening. It's mostly grad students, postdocs, faculty, maybe a few undergrads here and there. Are there openings in your lab or other areas for people interested in this type of work? And if so, where should they go to learn more or see about getting involved?
Well, I think that for the people interested, there are a lot of groups now around the world who work on this. The field is basically expanding very, very quickly. We have constantly new positions, of course, also because of the interest in the work that is expanding. There are also several colleagues of us that are working. There will be also, for those interested, a Keystone Conference on innate immune memory / trained immunity next year in Banff in Canada in March. So for those who are really interested in the field, that is the place to be, basically, because all the major groups involved in the field will be there, and I think it will be very exciting meeting. And I think there will be a lot of possibilities to work in this in the coming years.
Great. Then the field looks very promising, and we're excited to hear more about future research. I think that we are reaching the end of our conversation here. But we like to ask at the end kind of a question a little more personal to get to know our guests. And so I guess that one of the questions I have for you is, what is a hobby you would have liked to pursue but never had the time for?
A hobby that I would like to pursue. I mean, I have been always passionate by history, for example, and I always dreamed, let's say, to join at a certain moment an archaeological site to stay for a couple of weeks, for example, to discover a new civilization in the Middle East, for example, or wherever exciting things are happening. And that is something that I would have liked to do. But of course, that was tough with all the other things.
Yeah, that sounds very exciting. I imagine like beginning of the century, English explorers, just going to middle of, I know, Egypt to find some random tomb.
Yes, exactly. That would have been.
And bringing the things back in big craters full of hay and just some basis or something.
Now they're using sonar to find all of this stuff underground that no one ever realized was there, which is pretty fascinating. So maybe you can still do that as a second career.
Well, I am involved, actually. I am involved with colleagues. I'm from Romania originally myself, so I know a lot of historians from Romania, and I am a little bit involved in some work on ancient DNA from Neolithic and Bronze Age in Romania. So it's still I'm still trying to pursue some of these things.
Sounds exciting.
My wife's family is from the border of Romania and Hungary, the part that kept going back and forth. Somewhere that like we looked at visiting, but you'd have to like fly in and then take a train and then take a bus to like the most remote place in Rome. I'm like, I have to look up the name of it, but it's quite like in the corner. But that's where her family was from before they ended up coming here after the war.
Well, there is your Romanian connection, Jason. Anyway, it's been great talking to you. Thank you so much for joining us for this conversation. And we wish you a lot of success with your upcoming research. And we wish you a great day.
Thank you very much.
Thank you very much for the invitation. It was a pleasure. Thank you.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and roundup papers. You can also reach out to us on Twitter at @ImmunoPodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.